Hello there, and welcome to the Sleepy Science Society. Tonight, we're drifting into one of Earth's most extraordinary and quietly thriving worlds, a place where frozen seawater becomes home to an entire ecosystem of remarkable life. As you settle in, I invite you to like and subscribe if you enjoy these peaceful journeys through science. Your support helps other curious minds find this corner of calm exploration. For now, simply let yourself relax. Allow your breathing to slow. Let the warmth of curiosity wrap around you as we venture together into the hidden universe of sea ice ecosystems. Imagine standing at the edge of the polar ocean, where the boundary between water and sky blurs into a vast expanse of white. Beneath your feet lies not just frozen water, but an entire living world. Within the crystal lattice of ice, microscopic channels pulse with life. Algae bloom in golden brown carpets. Bacteria thrive in conditions that would destroy most organisms. Tiny crustaceans navigate labyrinths of frozen brine. Fish shelter in the shadowed caves beneath ice floes. Seals rest on the surface while their pups nurse in snow dens carved from the frozen landscape. This is the sea ice ecosystem, and it exists in both the Arctic and Antarctic regions of our planet. These frozen realms might seem barren at first glance, silent and still under endless winter darkness or the glare of midnight sun. Yet they harbor some of the most productive ecosystems on Earth, supporting food webs that stretch from microscopic bacteria to the largest whales. The ice itself becomes architecture, scaffolding, nursery, and hunting ground all at once. What makes these ecosystems so fascinating is their paradoxical nature. Ice, which we think of as lifeless and sterile, becomes habitat. Salt water that freezes creates pockets of even saltier brine that somehow nurture delicate organisms. Darkness that lasts for months gives way to explosive blooms of life. Extreme cold that would kill most creatures instead shapes adaptations of breathtaking elegance. In the places where conditions seem most hostile to life, life finds ingenious ways not merely to survive, but to flourish. Tonight, we will explore this frozen world from its smallest inhabitants to its grandest. We will descend into microscopic channels within the ice where algae conduct photosynthesis in near darkness. We will meet the bacteria that thrive at temperatures well below the freezing point of pure water. We will follow tiny amphipods as they graze on algal forests growing upside down beneath the ice. We will watch as fish use the underside of ice floes as shelter and hunting grounds. And we will understand how changes in sea ice are reshaping these ancient ecosystems in ways that ripple across the entire planet. So, breathe deeply. Let your muscles soften. Allow yourself to drift into the wonder of these frozen seas, where the impossible becomes everyday reality, and where life has written one of its most remarkable chapters. The formation of sea ice, a frozen architecture. Before we can understand the ecosystem, we must understand the ice itself, for sea ice is far more than simple frozen water. Its structure, its composition, and its formation process create the very foundation upon which this entire ecosystem rests. The story of sea ice begins when autumn temperatures drop at the poles, when the angle of sunlight grows shallow, and when the ocean surface begins its transformation from liquid to solid. Seawater does not freeze the way fresh water does. Pure water freezes at 0 degrees Celsius, but ocean water contains dissolved salts, primarily sodium chloride, along with magnesium, sulfate, calcium, and potassium. These salts lower the freezing point to approximately minus 1.8 degrees Celsius. When temperatures drop below this threshold, something remarkable begins to happen. Ice crystals start to form on the ocean surface, but these crystals are not made of salt water. They are made almost entirely of pure water ice. As the ice crystals form, they exclude most of the dissolved salts. The salt molecules, along with other particles suspended in the water, get pushed out from the growing ice matrix. This process, known as brine rejection, creates pockets and channels of extremely salty water within and beneath the forming ice. Imagine tiny rivers of concentrated brine threading through a solid matrix of relatively pure ice crystals. This structure is absolutely critical to the ecosystem that will develop within the ice. The initial formation of sea ice occurs in several stages, each creating different types of ice with distinct characteristics. First, small, disc-shaped crystals called fragile ice form in the supercooled surface water. These delicate platelets look like slush or grease floating on the ocean surface, giving it an oily appearance that mariners have called grease ice. As temperatures continue to drop, these fragile crystals accumulate and consolidate, forming a continuous sheet of young ice called nylus. Nylus is thin, dark, and flexible, bending with the waves rather than breaking. As the ice continues to thicken, it becomes first-year ice. In the Arctic, first-year ice typically reaches thicknesses of about 1 to 2 meters by the end of winter. In the Antarctic, where conditions are somewhat different, first-year ice often remains thinner, frequently less than 1 meter thick. If this ice survives the summer melt season, it becomes second-year ice, and if it survives multiple summers, it becomes multi-year ice. Multi-year ice in the Arctic can reach thicknesses of 2 to 4 meters or more, with some ancient flows growing even thicker through the process of rafting, where ice sheets collide and pile atop one another. The physical structure of sea ice is extraordinarily complex. At the macroscopic scale, sea ice consists of a mosaic of individual ice flows separated by cracks called lids and persistent open water, areas called polynyas. These features create dynamic landscapes where ice is constantly in motion, grinding against neighboring flows, opening and closing channels of water, and reshaping the surface through pressure ridges where colliding ice buckles upward into jagged formations. But it is the microscopic structure of sea ice that truly matters for its role as habitat. Within the solid ice matrix exists an intricate, three-dimensional network of brine channels and pockets. These features form because as the ice freezes, the excluded salt creates regions of highly concentrated brine that remain liquid even at temperatures well below the freezing point of seawater. The channels range in diameter from a few micrometers to several millimeters, creating a labyrinth of interconnected passages throughout the ice. The size and connectivity of these brine channels depend on temperature. As winter deepens and the ice grows colder, the brine channels narrow and the salinity of the brine increases further. Some pockets may become so salty that they remain liquid at temperatures as low as minus 20 degrees Celsius or even colder. When spring arrives and temperatures rise, the brine channels expand again, becoming more interconnected and allowing greater movement of organisms and nutrients through the ice matrix. This brine channel network typically occupies between 10 and 30% of the total volume of sea ice. Imagine a block of ice the size of a refrigerator. Now, imagine that within that block, roughly a quarter of the space consists of liquid channels filled with brine several times saltier than normal seawater. This is the environment that sea ice organisms must inhabit, and it shapes every aspect of their biology. The vertical structure of sea ice also matters immensely. The uppermost layer consists of relatively pure ice and, during winter, a layer of snow. This snow cover plays a crucial role in the ecosystem by insulating the ice beneath, maintaining warmer temperatures in the lower ice layers, and by blocking much of the incoming sunlight. Below the snow and surface ice lies the interior ice where brine channels are most abundant. At the very bottom, where the ice meets the liquid ocean, conditions are most hospitable for life. This lowest region, typically the bottom 10 to 20 centimeters of ice, is where temperatures remain close to the freezing point of seawater, where brine channels connect directly to the nutrient-rich ocean water below, and where the bulk of the ice ecosystem concentrates. The optical properties of sea ice add another layer of complexity. Snow-covered ice reflects most incoming sunlight, with albedo values often exceeding 80 to 90%. This means that very little light penetrates through the ice to support photosynthesis. However, as spring progresses and snow melts, forming melt ponds on the ice surface, light transmission increases dramatically. These melt ponds, with their darker, thinner ice and water-filled surfaces, can transmit 10 to 40 times more light than snow-covered ice. This transformation from opaque white surface to translucent blue pools marks a fundamental shift in the ecosystem, triggering blooms of ice algae and setting in motion the productive spring season. Sea ice also acts as a physical barrier between the ocean and atmosphere, with profound effects on both. It insulates the ocean from the cold polar air, reducing heat loss from the water below. It prevents wind from generating waves on the ocean surface, creating calmer conditions beneath the ice. It provides a physical platform for animals ranging from microscopic organisms to polar bears and penguins. And when it melts, it releases fresh water into the surface ocean, reducing salinity and creating a stable, stratified water column that favors phytoplankton blooms. Understanding the formation and structure of sea ice reveals why it becomes such vital habitat. The brine channels provide liquid environment in an otherwise frozen world. The connection to nutrient-rich seawater at the ice bottom ensures food availability. The physical stability of the ice platform creates refugia from predators and from the turbulent ocean. The seasonal cycle of freezing and melting drives productivity rhythms that synchronize entire food webs. Sea ice is not simply frozen ocean. It is a complex, dynamic structure that creates unique ecological opportunities found nowhere else on Earth. brine channels, rivers of salt. Within the ice, within the frozen matrix of sea ice, exists a hidden network that makes life possible in one of Earth's most extreme environments. These are the brine channels, microscopic rivers of super salty water that thread through the ice like veins through tissue. To understand the sea ice ecosystem, we must first descend into these tiny passages and comprehend both their physical nature and their role as habitat for the organisms that call them home. When seawater begins to freeze, physics and chemistry conspire to create something unexpected. As ice crystals form, they consist almost entirely of frozen water molecules arranged in a regular crystalline lattice. This lattice structure cannot easily accommodate salt ions, so as the ice grows, it excludes the dissolved salts. These rejected salts accumulate in the spaces between ice crystals, creating pockets of liquid brine that can be several times saltier than normal seawater. In the coldest winter conditions, brine within sea ice can reach salinities exceeding 100 parts per thousand, compared to the typical ocean salinity of about 35 parts per thousand. The formation of brine channels follows a fascinating developmental sequence. When ice first forms, the excluded brine exists in disconnected pockets scattered throughout the ice matrix. As freezing continues, these pockets become more organized. Gravity causes denser brine to sink, creating vertical drainage channels. At the same time, temperature gradients within the ice drive brine migration, with warmer regions melting slightly and colder regions freezing further. The result is a complex, three-dimensional network of channels, pockets, and interconnected passages that evolves constantly as temperature and salinity conditions change. In winter, when temperatures plunge well below freezing, the brine channels become narrower and less connected. The brine itself grows more saline as more ice forms around it, concentrating the dissolved salts into ever smaller volumes. During the darkest, coldest months, some of the smallest channels may contain brine so concentrated that it approaches saturation for various salts. These extreme conditions would seem to preclude life, yet organisms not only survive here but remain active, albeit at reduced metabolic rates. The diameter of brine channels varies tremendously depending on ice age, temperature, and location within the ice. The smallest channels measure only a few micrometers across, barely wide enough for a single bacterial cell to pass through. The largest can reach several millimeters, spacious enough to accommodate small crustaceans and complex algal filaments. Most channels fall somewhere in between, measuring tens to hundreds of micrometers in diameter. For reference, a human hair is typically about 70 micrometers in diameter. So, many brine channels are comparable to or smaller than the width of a hair. These dimensions create a claustrophobic world for the organisms living within. A copepod larva, only half a millimeter long, might seem tiny by human standards. Yet, within the brine channel network, it would be a giant, barely able to squeeze through the larger passages. Most life in the brine channels consists of organisms measured in micrometers: single-celled algae, bacteria, protozoans, and the microscopic larvae of larger animals. Size imposes a strict filter on which organisms can colonize the interior ice environment. The connectivity of the brine channel network profoundly influences the ecosystem within. When channels are well connected, organisms can move between different parts of the ice. Nutrients can flow from the ocean below into the interior ice, and waste products can be flushed away. Enhanced connectivity also allows larger organisms from the ocean to enter the ice temporarily, grazing on algae or seeking shelter. During spring and summer, as temperatures rise and ice melts from within, the brine channels expand and merge, creating an increasingly open network that peaks in connectivity just before the ice begins to disintegrate. In contrast, when winter cold constricts the channels, connectivity decreases. Isolated pockets of brine become cut off from the main network, trapping any organisms within. These isolated communities must survive on whatever nutrients remain in their tiny pockets, with no inflow of fresh resources from the ocean. It is a precarious existence, yet studies have shown that even in the most isolated brine pockets, bacteria and some algae species manage to persist, entering states of dormancy or reduced activity until conditions improve. The chemistry of brine channels extends beyond just high salinity. The concentrated salt solutions alter the freezing point depression, allowing liquid water to exist at temperatures far below 0 degrees Celsius. But other substances also concentrate in the brine. Dissolved nutrients such as nitrogen compounds, phosphates, and silicates become enriched as water freezes out. Dissolved organic matter from algal excretions and bacterial activity accumulates. Even dissolved gases like oxygen and carbon dioxide concentrate in the brine, though oxygen levels can drop to very low values in isolated pockets where biological respiration consumes it faster than it can be replenished. Perhaps most remarkably, recent research has revealed that organisms living in brine channels actively modify their habitat to make it more livable. Ice algae, bacteria, and other organisms produce extracellular polymeric substances, essentially biological slimes and gels, that alter the physical properties of the ice around them. These substances can prevent the complete closure of brine channels in cold conditions, inhibit the formation of sharp ice crystals that might damage cell membranes, and even influence the microstructure of the ice matrix itself. Life does not simply accept the conditions of the brine channels, it shapes those conditions to improve its own survival. The thermal environment within brine channels follows complex patterns. At the top of the ice, temperatures closely track air temperature, which can plunge to -40 degrees Celsius or colder in winter. At the bottom of this ice, where it contacts the ocean, temperatures remain near the freezing point of seawater, approximately -1.8 degrees Celsius. This creates a temperature gradient through the ice, with the coldest conditions at the surface and the warmest at the base. Organisms inhabiting different depths within the ice experience very different thermal environments, and the vertical distribution of life within the ice reflects these temperature differences, with the greatest biomass concentrated in the warmer, more hospitable bottom layers. Light penetration into brine channels represents another critical factor. In winter, when thick snow covers the ice surface, virtually no light reaches the brine channels. The organisms living within exist in near total darkness, unable to photosynthesize. living off stored energy or organic matter produced during the previous growing season. As spring arrives, snow cover thins and eventually melts, allowing sunlight to penetrate. Even then, light levels within brine channels remain dim, often less than 1% of surface illumination. Algae living here must be exquisitely adapted to function in these twilight conditions, extracting every possible photon to power their growth. The brine channel network also serves as a highway for biological materials. When ice algae photosynthesize, they excrete dissolved organic compounds that diffuse through the brine. Bacteria living nearby consume these compounds, converting them back into nutrients that the algae can use. Dead cells and waste products sink through the channels, accumulating in the lowest layers of ice or falling into the ocean below. In spring, when the channels expand and connect, zooplankton from the ocean can enter the ice briefly, grazing on algae before retreating back to the water. This constant flow of organisms, nutrients, and organic matter makes the brine channel network not just habitat, but also a conduit connecting the ice ecosystem to the ocean ecosystem below. During the spring melt season, the brine channel network undergoes dramatic transformation. Rising temperatures cause ice to melt from within, expanding channel size and creating new connections. Fresh meltwater flushes through the channels, lowering salinity and sometimes creating rapid flows that drain downward into the ocean. This flushing event can transport massive amounts of organic material accumulated over months from the ice into the water column below in a matter of days. For organisms living in the water, this pulse of food represents a bonanza, fueling zooplankton reproduction and attracting fish and larger predators. The brine channels thus represent far more than simple structural features of sea ice. They are dynamic habitats that change constantly with temperature, season, and icy age. They concentrate life while simultaneously imposing extreme conditions. They connect the frozen world of ice to the liquid world of ocean. They provide shelter, sustenance, and the physical space necessary for an ecosystem to exist within what appears from the outside to be lifeless, frozen seawater. These hidden rivers of salt are the arteries through which the lifeblood of the sea ice ecosystem flows, the microscopic pioneers. Ice algae and primary production. At the foundation of every sea ice ecosystem lie the microscopic algae that conduct the seemingly impossible task of photosynthesizing within and beneath frozen seawater. These organisms, collectively called ice algae or sympagic algae, are the primary producers that transform sunlight and dissolved nutrients into the organic matter that fuels entire food webs. Their story is one of remarkable adaptation to one of Earth's most challenging environments. Ice algae consist primarily of single-celled organisms, with diatoms dominating most Arctic and Antarctic sea ice communities. Diatoms are a diverse group of algae characterized by their beautiful glass-like cell walls made of silica, called frustules. Under a microscope, these frustules display geometric patterns of extraordinary complexity and elegance, forming boxes, cylinders, stars, and chains that look more like the work of master jewelers than of microscopic organisms. Species common in sea ice include pinnate diatoms like Nitschia frigida, Fragilariopsis species, and the remarkable Melocera arctica, which forms long filaments that can dangle several meters below the ice surface. But diatoms are not alone. The ice algae community also includes dinoflagellates, single-celled organisms with whip-like flagella that allow them to swim through the brine channels. Green algae and other groups also occur, though typically in smaller numbers. This diverse assemblage of microscopic plants faces a common challenge: how to grow and reproduce in an environment that seems designed to prevent photosynthesis. The first obstacle is light. In winter, polar regions experience months of continuous darkness. No sunlight means no photosynthesis, forcing ice algae to survive in a dormant state, living off stored energy and hoping to endure until the light returns. When spring finally arrives and the sun climbs above the horizon, another problem becomes apparent. The ice surface is typically covered by a thick layer of snow, which reflects and absorbs most incoming sunlight. What little light penetrates the snow must then pass through the ice itself, which scatters and absorbs more illumination. By the time light reaches the bottom of the ice where most algae concentrate, intensity has dropped to less than 1% of surface levels, sometimes as low as 0.1%. Ice algae have evolved remarkable adaptations to function in this perpetual twilight. They pack their cells with high concentrations of chlorophyll and other photosynthetic pigments, allowing them to capture nearly every available photon. They adjust their pigment ratios to match the spectral quality of light filtering through the ice, maximizing efficiency. Some species show adaptations to low light that rank among the most extreme ever documented in photosynthetic organisms, able to grow at light intensities that would starve most plants. The second major challenge is salinity. As we learned earlier, brine within sea ice can reach extremely high salt concentrations, sometimes exceeding 100 parts per thousand. Cells contain relatively fresh water compared to their environment, so osmotic pressure constantly tries to draw water out of their cells, threatening to dehydrate them. Ice algae combat this through osmoregulation, producing specialized organic compounds called osmolytes that increase the solute concentration within their cells, balancing the osmotic pressure and preventing water loss. One particularly important osmolyte is dimethylsulfonio-propionate, or DMSP. This compound not only helps with osmoregulation, but also protects cellular structures from damage caused by salt stress and freezing. When ice algae are damaged or die, enzymes break down DMSP into dimethyl sulfide, or DMS, a gas that escapes from the ice and ocean into the atmosphere. DMS plays a significant role in global climate because it serves as a nucleus for cloud formation over the polar oceans, influencing weather patterns and even the amount of sunlight reflecting back into space. Temperature represents another challenge, though perhaps a less severe one than light or salinity. Ice algae grow most abundantly in the bottom layers of sea ice where temperatures hover near minus 1.8 degrees Celsius, the freezing point of seawater. This is cold enough to slow molecular reactions dramatically, yet ice algae maintain active metabolism at these temperatures. They produce anti-freeze proteins and ice-binding proteins that protect their cell membranes from ice crystal damage and prevent intracellular freezing. Their enzymes are specially adapted to function efficiently at low temperatures, with different molecular structures than the equivalent enzymes in organisms from warmer environments. Nutrient availability might seem like a limitation, but surprisingly, ice algae often experience abundant nutrients. The ocean water immediately beneath the ice is typically rich in nitrogen, phosphorus, and silica, nutrients that rise from depth during winter mixing or arrive with currents. As long as the brine channels remain connected to this nutrient-rich water below, the algae have access to the raw materials they need for growth. The brine channels themselves can actually concentrate nutrients as water freezes out, creating locally enriched conditions. The combination of adequate nutrients and the stability provided by ice as a substrate allows ice algae to achieve remarkably high densities, sometimes forming visible brown or green layers on the underside of ice. The seasonal cycle of ice algae follows a predictable pattern driven by light availability. In early winter, as darkness descends and ice forms, algae living in the water column become trapped in the forming ice. These pioneering cells survive through the dark months in a dormant or semi-dormant state. Metabolic activity reduced to minimal levels, waiting for the return of sunlight. Some species can survive 5 to 6 months of continuous darkness. As spring approaches and light levels begin to rise, the algae sense the increasing illumination and resume active growth. The spring bloom of ice algae represents one of the most important biological events in polar ecosystems. As soon as sufficient light penetrates the ice, algae begin dividing, doubling their populations every few days under favorable conditions. This exponential growth transforms the bottom of the ice from a pale, nearly transparent layer into a dense carpet of golden brown or greenish cells. Biomass can increase by factors of 100 or 1,000 over a period of just weeks, creating concentrations of photosynthetic organisms far exceeding those found in the open water. This productivity is extraordinary. Studies have shown that ice algae can contribute anywhere from 10 to 57% of total primary production in ice-covered seas depending on the region and season. In areas of perennial ice cover where the overlying ice prevents phytoplankton growth in the water column, ice algae may provide the majority of all photosynthetic production. They achieve this despite occupying only the thin bottom layer of ice, a testament to their efficiency in utilizing the limited light and nutrients available. The organic matter produced by ice algae takes several forms. Some remains within the algal cells themselves, forming the basis for growth and reproduction. Some gets excreted as dissolved organic compounds that leak from healthy cells or are released when cells die. These dissolved organics support bacterial communities living in close association with the algae. Longer carbon chains and gel-like substances form the extracellular polymeric substances mentioned earlier, which alter ice structure and create a protective matrix around the algae. Finally, dead algal cells, fecal pellets from grazers, and other particulate organic matter accumulates at the ice bottom or sinks into the water when released. As spring progresses into summer and the ice begins to melt, The fate of ice algae diverges along different paths. Some cells remain trapped in the melting ice until they fall into the ocean as the ice disintegrates. Some actively sink, their dense silica cell walls carrying them down toward the sea floor, where they provide food for bottom-dwelling organisms. Some are consumed by zooplankton, grazing on the ice bottom. And some seed the phytoplankton blooms that develop in the surface water after the ice melts, taking advantage of the stable, nutrient-rich conditions created by ice melt, and continuing their growth in the open water. The taxonomic composition of ice algae communities varies between the Arctic and Antarctic, and even between different regions within each pole. Arctic ice algae tend to be dominated by pinnate diatoms, particularly species adapted to life within the brine channels and on ice under surfaces. Antarctic communities show greater diversity, with significant populations of dinoflagellates in addition to diatoms. This biogeographic variation reflects the different evolutionary histories and environmental conditions of the two polar regions, reminding us that while sea ice ecosystems share general features, each region has developed its unique biological character. Ice algae represent the engine that powers sea ice ecosystems. Without their ability to capture solar energy and transform inorganic nutrients into organic matter, the complex food webs we will explore could not exist. These microscopic pioneers, conducting photosynthesis in frozen twilight, creating life from light and dissolved minerals, stand as testimony to the extraordinary versatility and persistence of life on Earth. The hidden residents, bacteria, viruses, and protozoans. While ice algae capture much attention as the primary producers of sea ice ecosystems, they share their frozen habitat with an extraordinary diversity of even smaller organisms. Bacteria, archaea, viruses, and heterotrophic protozoans exist in numbers that dwarf algal populations, playing crucial roles in nutrient cycling, decomposition, and energy flow. These hidden residents represent the invisible majority of sea ice life, and understanding them reveals the ecosystem's true complexity. Bacteria inhabit every part of sea ice, from the surface down to the ice-ocean interface, from the smallest brine pockets to the largest channels. Bacterial abundances in sea ice often reach hundreds of millions to billions of cells per liter of melted ice, concentrations that can be 10 to 100 times higher than in the surrounding ocean water. This enrichment reflects both the trapping of bacteria during ice formation and their subsequent growth within the ice environment. Sea ice bacteria display remarkable physiological diversity, reflecting adaptations to multiple environmental stresses simultaneously. They must tolerate sub-freezing temperatures, sometimes reaching minus 20 degrees Celsius or colder in winter. They must cope with extreme salinities as brine concentrates in shrinking channels. They must survive near total darkness for months at a time. And when spring arrives, they must suddenly adjust to increased light, lower salinity from meltwater, and rapidly changing temperatures. Despite these challenges, bacterial communities in sea ice remain active throughout the year, though their metabolic rates slow dramatically during the coldest, darkest months. The functional roles of sea ice bacteria are diverse and essential. Heterotrophic bacteria consume dissolved organic matter released by algae, converting it back into nutrients like ammonium and phosphate that algae can reuse. This tight coupling between algae and bacteria creates efficient recycling loops that help maintain productivity even when external nutrient inputs are limited. Other bacteria specialize in decomposing dead cells and particulate organic matter, breaking down complex molecules into simpler compounds. Some bacteria are capable of unusual metabolic pathways that allow them to use alternative energy sources when organic matter is scarce, such as oxidizing sulfur compounds or using chemical energy from nitrogen transformation. Cryoprotective mechanisms allow bacteria to survive freezing conditions. Many species produce extracellular polymeric substances similar to those made by algae, creating protective slime layers that prevent ice crystal formation in their immediate vicinity. Others accumulate compatible solutes within their cells, organic compounds that protect proteins and membranes from freeze damage. Some bacteria produce anti-freeze proteins that bind to ice crystals and prevent their growth, while others adjust their membrane lipid composition to maintain fluidity at low temperatures. These adaptations represent evolution's solutions to the physics and chemistry of life at the freezing point. The bacterial community composition changes with season, ice age, and position within the ice. Winter communities differ substantially from summer communities, reflecting shifts in available nutrients, light, and temperature. Multi-year ice harbors different bacterial assemblages than first-year ice, suggesting that ice age influences the habitat characteristics in ways that select for particular bacterial groups. And bacteria in the surface ice layers differ from those at the ice bottom, with vertical zonation patterns that mirror the physical and chemical gradients within the ice. Recent research using molecular techniques has revealed that sea ice bacteria are far more diverse than previously recognized. Traditional culturing methods, which grow bacteria on laboratory media, recovered only a small fraction of the bacteria present. Modern DNA sequencing approaches have identified hundreds of distinct bacterial groups in sea ice samples, many never before cultured or characterized. This hidden diversity suggests complex ecological interactions and niche partitioning that we are only beginning to understand. Viruses add another layer of complexity to sea ice microbial communities. Viral abundances in sea ice are similarly elevated compared to seawater, sometimes reaching billions to tens of billions of virus-like particles per liter of melted ice. These viruses primarily infect bacteria and algae, serving as major agents of mortality for their hosts. When a virus infects a bacterial cell, it hijacks the cellular machinery to produce more virus particles, eventually causing the cell to burst open and release hundreds of new viruses to infect other cells. This process, called viral lysis, has profound implications for nutrient cycling and carbon flow in sea ice ecosystems. Viral infection of bacteria accelerates the conversion of particulate organic matter into dissolved organic matter. When bacterial cells lyse, their contents spill into the surrounding brine, releasing dissolved organic compounds, nutrients, and cellular fragments. These materials become available to other bacteria and algae, supporting the microbial loop and enhancing nutrient recycling. Models suggest that viral lysis might release 20 to 30% or more of bacterial and algal production back into the dissolved pool, fundamentally altering the fate of carbon and energy in the ecosystem. Viruses also influence community composition by selectively infecting particular host species. When one bacterial or algal species becomes very abundant, virus populations that infect that species increase in response, eventually reducing the host population and allowing other species to flourish. This kill-the-winner dynamic prevents any single species from dominating, maintaining diversity within microbial communities. The evolutionary arms race between hosts and viruses drives rapid adaptation, with hosts evolving resistance mechanisms and viruses evolving counter-strategies to overcome resistance. Heterotrophic protozoans represent the next size class above bacteria and viruses. These single-celled organisms include ciliates, flagellates, and other groups that feed by consuming bacteria and small algae. Protozoan abundances in sea ice can reach tens of thousands to hundreds of thousands per liter, with biomass sometimes comparable to or exceeding bacterial biomass. They serve as important grazers, controlling bacterial populations and channeling energy up the food web to larger organisms. Ciliates, characterized by hair-like cilia covering their surfaces, are particularly diverse and abundant in sea ice. Some species feed on bacteria by sweeping them into mouth-like structures with coordinated ciliary beating. Others engulf larger food particles, including algae and detritus. Ciliate diversity in sea ice includes both species that are cosmopolitan, found throughout the world's oceans, and others that appear to be specialized for sea ice habitats, rarely found elsewhere. Their presence indicates that sea ice supports a complex microbial food web with multiple trophic levels. Heterotrophic dinoflagellates add further complexity. Unlike their photosynthetic cousins, these dinoflagellates obtain energy by feeding on other organisms. Some species can engulf prey cells nearly as large as themselves, consuming algae and even smaller dinoflagellates. Others feed using tentacle-like structures that pierce prey cells and suck out their contents. The diversity of feeding strategies among sea ice protozoans reveals an ecosystem where multiple pathways exist for energy transfer from primary producers to higher trophic levels. The invisible residents of sea ice thus create a microbial world of enormous complexity and activity. Bacteria decompose organic matter and recycle nutrients at temperatures where most biochemistry would grind to a halt. Viruses infect hosts and redirect energy flows through pathways we have only recently begun to map. Protozoans graze on the bacterial populations, coupling microbial production to larger organisms. Together, these hidden communities process the organic matter produced by ice algae, maintaining the nutrient cycles that keep the ecosystem productive and forming the base of a food web that ultimately supports fish, birds, seals, and whales. Small grazers and crustaceans. Life at the ice-water interface. Rising above the microscopic world of bacteria and protozoans, we encounter organisms large enough to see with the naked eye, yet small enough to navigate the intricate architecture of sea ice. These are the metazoans, the multicellular animals that graze on ice algae, shelter in brine channels, and form crucial links between primary production and higher trophic levels. Among them, small crustaceans dominate, turning the underside of sea ice into a bustling foraging ground. Amphipods represent some of the most abundant and characteristic animals associated with Arctic sea ice. These small crustaceans, typically measuring 1 to 3 centimeters in length, have bodies compressed laterally and curved like commas. Several species are so closely associated with ice that they are considered sympagic, living their entire lives in, on, or under sea ice. Gamarus wilkitskii stands as perhaps the most iconic Arctic sympagic species, found crawling along the underside of ice floes throughout the central Arctic Ocean. Apharusa glacialis, Onissimus glacialis, and Onissimus nanseni represent other common sympagic amphipods, each with slightly different habitat preferences and feeding strategies. These amphipods have evolved remarkable adaptations for life at the ice-water interface. Their bodies remain flexible enough to squeeze through brine channels, allowing them to shelter within the ice when threatened by predators. They tolerate the sub-freezing temperatures and elevated salinities found near the ice. Many species display color changes, turning more transparent in summer when the ice is clearer, and darker in winter when the ice is more opaque, potentially helping them avoid detection by visual predators. Their life cycles are synchronized with the seasonal cycles of ice algae production, timing reproduction to coincide with the spring algal bloom when food is most abundant. The feeding behavior of ice amphipods plays a critical role in the ecosystem. They graze directly on the layers of ice algae coating the underside of ice floes, scraping cells from the surface with specialized mouthparts. This grazing can significantly reduce algal biomass in some areas, potentially controlling algal abundance and influencing species composition. However, amphipods are somewhat selective in their feeding, preferring certain algal species over others, which can alter community structure by favoring ungrazed species. Beyond direct grazing, amphipods influence the fate of organic matter through their fecal production. After consuming algae, they produce fecal pellets that are denser than seawater and sink rapidly. These pellets represent a significant pathway for carbon export from the ice to the ocean below and ultimately to the seafloor. Studies have shown that amphipod grazing can enhance the downward flux of particulate organic matter, accelerating the delivery of surface production to deep ecosystems. The fecal pellets also serve as food for other organisms, including bacteria, protozoans, and deposit-feeding animals on the seafloor. Amphipods themselves become prey for a variety of larger animals. Arctic cod feed extensively on ice amphipods, especially the juveniles. Seabirds diving beneath the ice capture amphipods near the ice-water interface. Even some seals include amphipods in their diets, though they represent a minor component compared to fish. This positions ice amphipods as crucial intermediaries, transferring energy from microscopic algae to fish and birds that are too large to feed on algae directly. In the Antarctic, the ecological role played by amphipods in the Arctic is largely taken by euphausiids, commonly known as krill. Antarctic krill, Euphausia superba, is perhaps the most famous, forming enormous swarms in the Southern Ocean that serve as the primary food source for whales, seals, penguins, and countless other species. While adult Antarctic krill are predominantly pelagic, living in open water rather than in association with ice, juvenile stages often shelter under and within sea ice. Another species, Euphausia crystallorhophias, shows stronger association with ice throughout its life cycle. Ice-associated krill feed on ice algae much like Arctic amphipods, grazing on the underside of ice floes or filtering algae from the water. They possess sophisticated filtering appendages that allow them to capture even small algal cells efficiently. Observations by researchers working beneath Antarctic sea ice have documented krill swimming upside down, scraping algae from the ice bottom with specialized movements, essentially harvesting the algal carpet that develops there each spring. The importance of sea ice to Antarctic krill populations extends beyond food. Ice provides refuge for juvenile krill, protecting them from predators that hunt in open water. The complex, three-dimensional structure of ice undersides, with its ridges, cracks, and cavities, offers hiding places where small krill can escape larger predators. This protective function may be critical for krill recruitment, determining how many juveniles survive to adulthood and thus influencing the abundance of krill available to support the larger Antarctic ecosystem. Copepods represent another abundant group of small crustaceans associated with sea ice, though their relationship with ice tends to be more facultative than the obligate association seen in sympagic amphipods. Calanoid copepods, particularly species like Calanus glacialis in the Arctic, perform remarkable daily vertical migrations that bring them into contact with sea ice. During the polar spring, when ice algae are blooming but phytoplankton in the water column have not yet developed, copepods ascend from depth to feed on the ice algae accumulating at the ice-ocean interface. They feed during periods when algal productivity is high, then descend to deeper, darker waters to digest their meals and avoid predators. This behavior creates a vital connection between the icy ecosystem and the deeper ocean. Copepods feeding on ice algae transport the energy and nutrients fixed by those algae down into the water column when they descend. Some of this material is respired by the copepods, releasing carbon dioxide at depth and contributing to the biological carbon pump that stores carbon in the deep ocean. Some is excreted as fecal pellets that sink even deeper. And some remains stored in copepod bodies that may themselves become food for fish or other predators. Larvae of various invertebrates also occupy the ice-water interface during particular seasons. Polychaete worms, mollusks, echinoderms, and other bottom-dwelling animals often have planktonic larval stages that drift in the water column. In shallow Arctic seas, these larvae sometimes enter the brine channel network at the ice bottom, feeding on the rich algal and bacterial communities there while enjoying some protection from pelagic predators. When they complete their larval development, they settle to the seafloor and metamorphose into their adult forms, having used sea ice as a temporary nursery habitat. Even truly microscopic metazoans find homes in sea ice. Rotifers, tiny animals with crown-like bands of cilia around their mouths, occur in brine channels where they feed on bacteria and small algae. Turbularian flatworms, some barely visible to the naked eye, glide through the brine hunting for protozoans and bacteria. Nematode worms, relatives of the more familiar soil nematodes, inhabit sea ice in surprising numbers, occupying a similar ecological niche to their terrestrial cousins by feeding on bacteria and participating in decomposition processes. The community of small grazers and crustaceans associated with sea ice reveals an ecosystem of remarkable richness. Multiple species coexist by partitioning resources, some feeding primarily on algae, others on bacteria or detritus, some remaining always at the ice surface, others moving between ice and open water. Competition is moderated by spatial heterogeneity in the ice environment, which creates countless microhabitats, each slightly different in temperature, salinity, food availability, or physical structure. Predation is ever present, driving behaviors like sheltering in brine channels and coordinating activity patterns with the daily cycle of predator movement. These small animals represent the critical middle links in sea ice food webs. They consume the vast production of ice algae and bacteria, converting microscopic biomass into larger packages that can be consumed by fish, birds, and mammals. They process organic matter through their guts, breaking it down and making nutrients available for recycling. They support the spectacular abundance of larger animals for which polar ecosystems are famous. Without the amphipods, krill, copepods, and myriad smaller metazoans that thrive at the ice-water interface, the complex ecosystem of sea ice could not function. Fish, seals, and the larger inhabitants, as we ascend the food web, from microscopic algae through small crustaceans, we arrive at the larger animals that capture human imagination when we think of polar seas. These are the fish that school beneath the ice, the seals that haul out onto ice floes to rest and give birth, the polar bears and penguins that depend on ice as their primary habitat. These charismatic animals could not exist without the foundation provided by the organisms we have already explored. Yet, they define the character of sea ice ecosystems in ways that smaller creatures cannot. Arctic cod, Boreogodus saida, occupies a unique position in Arctic marine food webs as the dominant fish species associated with sea ice. This small fish, rarely exceeding 30 centimeters in length, is exquisitely adapted to life in ice-covered waters. Arctic cod tolerate sub-freezing temperatures, produce antifreeze glycoproteins in their blood that prevent ice crystal formation, and maintain active swimming and feeding behavior throughout the winter, when most other fish become sluggish. Their association with sea ice is so strong that they are often found swimming in dense schools directly beneath ice floes, feeding on ice amphipods, copepods, and other zooplankton. Juvenile Arctic cod rely heavily on sea ice as nursery habitat. Young fish shelter in the complex underside structure of pressure ridges, in spaces between ice blocks and along the edges of ice flows. This ice-associated habitat provides protection from larger predators, while offering abundant food in the form of small crustaceans grazing on ice algae. The survival of juvenile Arctic cod appears to be closely linked to the availability and condition of sea ice, with recruitment success varying in response to changes in ice cover. The importance of Arctic cod extends far beyond its own ecological role. This species serves as a crucial forage fish, transferring energy from zooplankton to a vast array of predators. Ringed seals feed extensively on Arctic cod, particularly during the winter and spring. Seabirds, including guillemots, kittiwakes, and Arctic terns, prey on cod when they are available near the surface. Even polar bears occasionally catch and eat Arctic cod when seals are scarce. The cod's position linking zooplankton production to marine mammals and birds makes it a keystone species in Arctic food webs, and anything that affects cody populations ripples through the entire ecosystem. In the Antarctic, the ecological niche filled by Arctic cod in the north is occupied by Antarctic silverfish, Pleurogramma antarctica. This species shows similarly strong associations with sea ice, particularly during its early life stages. Antarctic silverfish spawn on the underside of sea ice, attaching their eggs to the ice where they develop until hatching. The larvae remain associated with ice, feeding on copepods and other zooplankton concentrated at the ice-water interface. As they grow, silverfish continue to use ice as habitat, though adult fish also inhabit the open water of the Southern Ocean. Antarctic silverfish constitute the primary prey for many Antarctic predators. Adelie penguins feed almost exclusively on silverfish during certain seasons. Weddell seals include silverfish in their diets. Numerous seabirds prey on them when they are available. The close association between silverfish and sea ice means that changes in ice conditions can have cascading effects on the many species that depend on silverfish for food, making this small fish another keystone species whose importance far exceeds what its modest size might suggest. Seals represent the pinnacle of mammalian adaptation to ice-covered seas. Different seal species have evolved distinct strategies for exploiting sea ice habitat, creating a guild of ice-associated predators that partition resources and avoid direct competition. Ringed seals, the smallest Arctic seals, remain closely associated with ice throughout the year. In winter, they maintain breathing holes through the ice, gnawing away at the ice from beneath to keep openings clear even as ice thickens. Female ringed seals excavate birth layers in snowdrifts on the ice, creating sheltered chambers where they give birth and nurse their pups. The survival of ringed seal pups depends critically on snow conditions. The birth layer provides insulation from the extreme cold of the Arctic winter and concealment from predators, particularly polar bears. When snow depth is insufficient, or when rain-on-snow events create hard, icy crusts that mothers cannot excavate, seal reproductive success plummets. Climate change is altering snow conditions on Arctic sea ice, creating concerns about the future of ringed seal populations and the polar bears that depend on them. Bearded seals, larger than ringed seals, prefer areas of broken ice over relatively shallow water, where they can dive to the seafloor to feed on bottom-dwelling invertebrates. Harp and hooded seals are highly migratory, moving with the seasonal ice advance and retreat. They give birth on stable ice floes during spring, with massive aggregations of mothers and pups gathering on the ice in traditional breeding areas. These aggregations provide spectacular wildlife displays, but also make the seals vulnerable to predation and disturbance. In the Antarctic, Weddell seals occupy a niche similar to Arctic ringed seals, remaining associated with fast ice along the Antarctic coast year-round. They too maintain breathing holes through thick ice, using their teeth to keep openings clear. Crab-eater seals, despite their name, feed primarily on krill rather than crabs, and are strongly associated with pack ice. Leopard seals patrol ice edges, preying on penguins, other seals, and fish. Ross seals occupy the dense, heavy pack ice of the Antarctic interior, while Antarctic fur seals and southern elephant seals are less dependent on ice, hauling out on beaches and rocky shores rather than ice floes. The feeding ecology of ice-associated seals reveals the importance of the ice ecosystem in supporting these large predators. Ringed seals feed on Arctic cod and other fish, as well as larger crustaceans like amphipods. Crab-eater seals filter krill from the water using specially adapted teeth. Leopard seals are generalist predators, taking prey ranging from krill to penguins to other seal species. All these seals ultimately depend on the productivity originating with ice algae, channeled through zooplankton and fish to reach these top predators. Polar bears stand as the apex predator of Arctic sea ice ecosystems, and perhaps the most ice-dependent of all mammals. Polar bears hunt primarily on sea ice, preying on ringed and bearded seals that they catch at breathing holes or by breaking into birth layers. Their entire life cycle revolves around sea ice. Polar bears mate on the ice. Pregnant females den in snowdrifts on land or on stable ice. Cubs learn to hunt on the ice. And the bears spend most of their lives traveling across the ice in search of prey. The hunting strategy of polar bears is intricately tied to ice conditions. In spring, when ringed seal pups are born in snow layers on the ice, polar bears patrol the ice surface searching for lairs. Using their remarkable sense of smell, they detect seal lairs beneath the snow, then break through the roof of the lair to capture the pup and sometimes the mother. This spring period, when seal pups are abundant and vulnerable, represents the critical feeding season for polar bears. The energy reserves they accumulate during spring must sustain them through the rest of the year, when hunting success declines. As summer arrives and sea ice retreats, polar bears face difficult choices. Some follow the retreating ice northward, remaining in ice-covered waters throughout the summer. Others are stranded on a land where they fast, living off stored fat reserves until the ice reforms in autumn. The duration of the ice-free season is thus critically important for polar bear survival, particularly for pregnant females who must accumulate sufficient fat reserves to sustain themselves through denning and nursing cubs. In the Antarctic, penguins fill ecological roles somewhat analogous to polar bears in terms of their dependence on ice, though as prey rather than predators, and as flightless birds rather than mammals. Emperor penguins breed exclusively on Antarctic fast ice, gathering in massive colonies during the Antarctic winter. They endure some of the harshest conditions on Earth, standing on ice for months through winter darkness and blizzards while incubating eggs and raising chicks. Adelie penguins also depend on sea ice, using it as resting platforms and feeding extensively on krill and silverfish associated with ice edges. The relationship between penguins and sea ice is complex. While sea ice provides breeding habitat and influences the distribution of prey, extensive ice can also prevent access to foraging areas. Emperor penguins must walk long distances between their breeding colonies on fast ice and open water where they feed. If ice extent is too great, the journey becomes prohibitively long, reducing adult survival and chick-rearing success. Conversely, insufficient ice or early breakup can destroy breeding colonies before chicks are ready to fledge. Seabirds add another dimension to ice ecosystem biodiversity. Ivory gulls in the Arctic are strongly associated with pack ice, feeding on Arctic cod and invertebrates in leads and polynias. Black guillemots nest in rocky crevices near ice-covered waters and feed on Arctic cod and invertebrates in the Antarctic. Snow petrels nest on rocky outcrops near ice-covered seas, while Antarctic petrels feed in open waters and at ice edges. These birds transfer nutrients from marine to terrestrial systems when they nest on land, and their breeding success often depends on the availability of ice-associated prey. Whales, though not ice-dependent in the same way as seals or polar bears, also interact importantly with sea ice ecosystems. Bowhead whales in the Arctic feed on zooplankton concentrated along ice edges. Beluga whales hunt for Arctic cod beneath the ice and use ice cover to avoid predation by killer whales. In the Antarctic, blue whales, fin whales, and humpback whales migrate to the Southern Ocean in summer to feed on enormous swarms of krill associated with sea ice and its productive ice edge zones. The larger inhabitants of sea ice ecosystems thus represent the accumulation of energy and nutrients that originated with microscopic ice algae and flowed upward through multiple trophic levels. They serve as highly visible indicators of ecosystem health, their abundance and reproductive success reflecting conditions at lower trophic levels. And they capture human attention and concern in ways that microscopic algae never could, making them ambassadors for the conservation of sea ice ecosystems. Yet, they could not exist without the hidden foundations provided by algae, bacteria, amphipods, and the countless smaller organisms that transform frozen seawater into vibrant, productive habitat. Seasonal rhythms, from winter darkness to summer bloom. The sea ice ecosystem does not exist in steady state. Instead, it pulses with seasonal rhythms that drive dramatic changes in community composition, biological activity, and energy flow. Understanding these seasonal cycles reveals how sea ice ecosystems have adapted to some of the most extreme seasonal variation found anywhere on Earth, where months of complete darkness alternate with months of continuous light, and where temperatures swing across a range of 50 degrees Celsius or more. The polar winter brings conditions that seem incompatible with life. At the height of winter, the sun remains below the horizon for weeks or months, depending on latitude. In the central Arctic Ocean, Polar night lasts from late October through mid-February, nearly 4 months of continuous darkness. In the Antarctic, darkness persists for similar durations. During these dark months, photosynthesis becomes impossible. Ice algae cannot grow and must survive on stored energy reserves. The only organisms capable of growing are heterotrophs, bacteria, protozoans, and metazoans that feed on organic matter accumulated during the previous productive season. Winter temperatures plunge to extreme lows. Air temperatures in the central Arctic regularly drop below -30 degrees Celsius, sometimes reaching -40 degrees Celsius or colder. These frigid temperatures cool the ice itself, causing brine channels to constrict at salinity to increase as water freezes out. Within the smallest brine pockets, salinity can reach concentrations so high that they approach the solubility limits of salt. Many organisms respond by entering dormancy, reducing metabolic activity to minimal levels, and waiting for conditions to improve. Yet winter is not entirely dormant. Bacterial communities remain active, albeit at greatly reduced rates. They consume the dissolved organic matter that pervades the brine channel network, slowly recycling nutrients and maintaining the basic functioning of the microbial loop. Some heterotrophic protozoans continue grazing on bacteria, their populations sustained by this slow turnover of organic matter. Even metazoans like ice amphipods remain present, though they too reduce activity levels and likely rely on stored energy reserves more than active feeding. The ice itself continues to grow during winter. As heat escapes from the ocean into the atmosphere, the ice thickens from its base, incorporating any organisms living in the water immediately beneath the ice. This process traps bacteria, algae, and small zooplankton in the growing ice, ensuring that when spring arrives and conditions improve, a seedy population of organisms is already present within the ice matrix. The trapping process is not random. Organisms that can produce substances that prevent complete ice formation around them, or that actively move into brine channels as the ice forms, have better chances of surviving entrapment. The first hint of spring arrives with the return of sunlight. In the Arctic, the sun climbs back above the horizon in mid-February at high latitudes, though initially for only a few minutes each day. This marks the end of polar night, and although light levels remain low, the increasing illumination triggers responses in ice organisms. Algae sense the returning light and begin adjusting their physiology in preparation for renewed growth. They increase chlorophyll production, ready photosynthetic machinery, and start shifting from dormancy to active metabolism. The spring transition represents the most critical period in the annual cycle of sea ice ecosystems. As light intensifies and temperatures slowly rise, conditions become favorable for algal growth. But this growth faces constraints. Snow cover accumulated over the winter reflects most incoming sunlight. Only where the snow is thin or where wind has swept areas clear can sufficient light penetrate to the ice bottom to support photosynthesis. These early season blooms often begin in localized patches where conditions are most favorable, then spread as snow melts and light transmission improves. The initial growth of ice algae proceeds slowly. Cells divide perhaps once every few weeks, gradually increasing biomass from its winter low. But as conditions continue to improve, growth accelerates. By late April or early May in the Arctic, and October or November in the Antarctic, ice algae enter exponential growth. Phase. Cell division occurs every few days. Algal biomass doubles and redoubles, and the underside of the ice transforms from pale translucence to rich golden brown carpets visible to the naked eye. This spring bloom of ice algae represents one of the most productive biological events on Earth relative to the habitat area involved. In a matter of weeks, algal biomass can increase by factors of 100 or 1,000. Chlorophyll concentrations in the bottom ice reach values far exceeding those ever found in open ocean phytoplankton blooms. The dense algal mats sometimes become so thick that they limit further growth by self-shading, blocking light from reaching deeper layers of the algal community. The factors controlling the spring bloom are complex and vary regionally. Light availability is paramount. Blooms begin when snow melts enough to allow sufficient light penetration. Nutrient supply from the underlying ocean must be adequate. Blooms flourish where brine channels connect to nutrient-rich water below. Temperature plays a role as warmer conditions within the brine channels support faster growth rates. And ice dynamics matter. Stable ice provides consistent habitat, while ice that breaks up prematurely can interrupt the bloom. As the ice algae bloom, they attract grazers. Copepods ascend from depth to feed at the ice-water interface. Amphipods congregate beneath areas of high algal biomass, scraping cells from the ice surface. Krill in the Antarctic begin filtering algae from the water. This grazing pressure can significantly reduce algal standing stock, though the algae often grow fast enough to compensate for losses. The interaction between algae and grazers creates a dynamic system where neither completely controls the other, with the balance shifting in response to environmental conditions. The peak of the ice algae bloom typically occurs in late spring or early summer, just before the ice begins its summer disintegration. At this point, ice algae biomass reaches its annual maximum. Brine channels are fully expanded and interconnected, and grazing pressure is intense. The system operates at maximum productivity, with energy flowing rapidly from algae through grazers to fish and higher predators. This represents the most active phase of the sea ice ecosystem. As summer progresses, the ice begins to melt. Melt ponds form on the ice surface as accumulated snow turns to water and saturates the surface ice. These ponds create windows through which sunlight can penetrate more easily, but they also alter ice structure, disrupting brine channels and causing sections of the ice bottom community to slough off into the ocean. The melting process releases ice algae into the water column, where they may seed phytoplankton blooms in the surface ocean or sink to the seafloor where they feed bottom communities. The transition from ice algae bloom to water column phytoplankton bloom represents a succession process central to polar ecosystem productivity. As the ice breaks up, the stable, stratified water layer created by ice melt provides ideal conditions for phytoplankton growth. Nutrients are abundant from winter mixing and ice melt. Light levels are high with continuous daylight. The phytoplankton bloom that develops can rival the ice algae bloom in productivity, supporting massive populations of zooplankton that in turn support fish, seabirds, and marine mammals. The summer open water season is relatively brief at high latitudes, lasting only 2 to 3 months in much of the Arctic and even less in the Antarctic pack ice zone. During this time, biological productivity shifts from ice-based to pelagic systems. Phytoplankton dominate primary production. Zooplankton populations peak. Fish school in surface waters. Seabirds congregate to feed on abundant prey. Marine mammals migrate to polar seas to take advantage of the summer bounty. The ice that remains becomes less important ecologically, though species like polar bears and some seals continue to depend on it. As autumn approaches, temperatures drop, daylight wanes, and the cycle begins anew. The ocean surface cools to its freezing point, and ice begins to form. Organisms in the water column become trapped in the forming ice. Algae enter dormancy as light levels fall. Bacteria persist in the brine channels. Metazoans either become incorporated into the ice or retreat to deeper waters to overwinter. The system enters the winter quiescent phase, accumulating resources and preparing for the explosive growth that will follow when light returns in spring. This seasonal cycle repeats annually, driving the pulse of life in sea ice ecosystems. The timing and intensity of each phase vary from year to year in response to weather, ice conditions, and ocean state. But the overall pattern remains consistent, shaped by the fundamental constraints of polar seasonality. The organisms living in and around sea ice have adapted their life cycles to synchronize with these rhythms, timing reproduction, growth, and migration to coincide with favorable conditions. The seasonal cycle thus represents not just environmental variation that organisms must tolerate, but a predictable pattern that has shaped evolutionary adaptations and structured the ecosystem itself. Arctic versus Antarctic. Two polar worlds. Although we often speak of sea ice ecosystems as if they were uniform across the polar regions, the Arctic and Antarctic differ profoundly in geography, oceanography, ice characteristics, and biology. These differences create distinct ecosystems that, while sharing general features, function in different ways and face different challenges. Understanding these contrasts reveals that sea ice ecosystems are not a single phenomenon, but rather variations on a theme, shaped by local conditions and evolutionary history. The most fundamental difference lies in geography. The Arctic Ocean is semi-enclosed, surrounded by the landmasses of North America, Greenland, Europe, and Asia. It connects to the Atlantic through the Greenland Sea, and the Bering Strait provides limited exchange with the Pacific. This geography constrains circulation patterns, creates extensive shallow continental shelves, and produces relatively stable ice conditions over much of the Arctic basin. In contrast, the Antarctic sea ice forms around the edges of a frozen continent surrounded by the deep Southern Ocean, which is unbounded to the north and circulates continuously around Antarctica. These geographic differences have profound implications for ice dynamics. Arctic sea ice includes both first-year ice that forms each winter and melts each summer, and multi-year ice that persists for multiple years, growing thicker and more massive with age. Multi-year ice once dominated the central Arctic Ocean, forming stable platforms that could be 5 to 10 meters thick in places. Antarctic sea ice, by contrast, consists almost entirely of first-year ice that forms in autumn, reaches maximum extent in winter, then melts almost completely in spring and summer. Only in the Weddell Sea and a few other locations does significant multi-year ice persist in the Antarctic. The seasonal cycles of ice extent differ between the poles. Arctic sea ice reaches maximum extent in March, covering about 15 million square kilometers, then retreats to a minimum in September of about 4 to 5 million square kilometers in recent years. This represents a loss of roughly 2/3 of the winter ice cover. Antarctic sea ice reaches maximum extent in September, covering approximately 18 to 19 million square kilometers, then retreats to a minimum in February of about 3 to 4 million square kilometers. The proportional change is similar, but the Antarctic ice extent is larger at both maximum and minimum, reflecting the vast area of ocean surrounding Antarctica. These differences in ice persistence and dynamics influence the biological communities that develop. Arctic multi-year ice provides stable, long-term habitat that allows the development of specialized ice-obligate species. The sympagic amphipods described earlier are largely restricted to multi-year ice, where brine channel networks remain established across years. In the Antarctic, the seasonal nature of nearly all sea ice favors species with life cycles adapted to annual formation and decay. Many Antarctic ice organisms must either complete their life cycles within a single growing season or possess the ability to survive in the water column during the ice-free summer. The age of ice also affects algal community composition. Arctic multi-year ice tends to harbor different algal species than first-year ice, with some diatoms specializing in the more established brine channel networks of older ice. Antarctic ice, being predominantly first-year, develops communities dominated by species adapted to colonizing and growing rapidly in newly formed ice. These biogeographic patterns reflect not only the physical differences in ice, but also the different evolutionary histories of Arctic and Antarctic marine organisms. Oceanographic conditions differ as well. The Arctic Ocean receives massive inputs of fresh water from rivers draining the northern continents, creating a strong vertical stratification with a relatively fresh surface layer overlying denser, saltier deep water. This stratification inhibits vertical mixing, which can limit nutrient supply to surface waters but also creates stable conditions favorable for ice formation. The Southern Ocean around Antarctica lacks significant freshwater input from land, but ice melt in summer creates strong stratification. The Antarctic Circumpolar Current, the world's largest ocean current, continuously transports water around Antarctica, influencing ice formation and biology. The food webs of Arctic and Antarctic sea ice ecosystems show both similarities and differences. Both rely on ice algae as primary producers. Both support populations of grazing crustaceans. Both include fish species adapted to cold water and ice association, and both support marine mammal and seabird populations at higher trophic levels. However, the specific species filling these roles differ. Arctic cod and Antarctic silverfish occupy analogous niches but are unrelated species that evolved independently. Similarly, sympagic amphipods dominate grazing in the Arctic, while krill dominate in the Antarctic, despite both groups being crustaceans. One striking difference lies in the importance of krill. Antarctic krill, Euphausia superba, occurs in enormous abundance, forming swarms that can contain millions of individuals and extending for kilometers. These krill swarms support the most productive ecosystems on Earth, sustaining populations of whales, seals, and penguins that have few parallels in the Arctic. The Arctic lacks an equivalent keystone species of such overwhelming abundance. While copepods are abundant in Arctic waters, they do not form the massive aggregations characteristic of Antarctic krill, and Arctic food webs tend to be more diverse and less dominated by single species. The historical legacy of human impacts differs between the poles. The Arctic has experienced centuries of commercial whaling, sealing, fishing, and more recently oil and gas development. Indigenous peoples have inhabited the Arctic for millennia, hunting marine mammals and fish. These activities have influenced Arctic marine ecosystems, though many Arctic species have shown remarkable resilience. The Antarctic, by contrast, was largely unknown until the 19th century, experienced intense but relatively brief periods of sealing and whaling in the 19th and early 20th centuries, and has been protected by the Antarctic Treaty System since 1961. Current human impacts in the Antarctic come primarily from fishing and climate change rather than from resource extraction or permanent settlement. Climate change affects the two polar regions differently. The Arctic is warming at more than twice the global average rate, a phenomenon called Arctic amplification. Arctic sea ice extent has declined dramatically, losing about 40% of its summer minimum extent since satellite observations began in 1909. Multi-year ice has declined even more severely, with thick, old ice becoming increasingly rare. These changes are fundamentally altering Arctic marine ecosystems, with ice-dependent species facing habitat loss and temperate species expanding northward. The Antarctic has shown more complex responses to climate change. While the Antarctic Peninsula has experienced rapid warming and significant ice loss, overall, Antarctic sea ice extent actually increased slightly from the 70s through about 2015, though with high year-to-year variability. This seemingly paradoxical increase resulted from changes in wind patterns and ocean circulation. However, since 2016, Antarctic sea ice has shown dramatic declines, with record low extents in recent years, suggesting that Antarctic ice may be entering a new phase of climate-driven change. These differences between Arctic and Antarctic sea ice ecosystems remind us that while general principles apply across polar regions, each pole faces unique conditions and challenges. The organisms that inhabit Arctic ice have evolved under different pressures than those in the Antarctic, resulting in distinct communities with different ecological characteristics. The threats they face differ in timing and intensity, and the management and conservation approaches needed to protect these ecosystems must be tailored to regional circumstances rather than applied uniformly. Yet despite these differences, both Arctic and Antarctic sea ice ecosystems share fundamental similarities. Both depend on the remarkable ability of organisms to thrive in and around frozen seawater. Both support productive food webs based on ice algae primary production. Both face profound changes as climate warming alters ice conditions. And both represent irreplaceable components of Earth's biological diversity, harboring adaptations and ecological relationships found nowhere else on the planet. The food web. Connections across scales. Now we step back to view the sea ice ecosystem as an integrated whole, tracing the flow of energy and nutrients from their initial capture by algae through the multiple pathways that connect all organisms in a complex web of feeding relationships. This food web perspective reveals how seemingly separate components of the ecosystem interact, depend upon one another, and together create a system whose emergent properties exceed the sum of its parts. At the base of every sea ice food web lie the primary producers, predominantly ice algae, but also including photosynthetic bacteria that contribute small amounts to primary production. These organisms capture energy from sunlight and use it to convert carbon dioxide and dissolved nutrients into organic matter. The rate of this primary production varies enormously with season, ice conditions, and location. But during peak bloom periods, ice algae can produce as much organic matter per unit area as the most productive terrestrial ecosystems. This represents the energy input that must support all other organisms in the ecosystem. The first major pathway from primary producers to consumers runs through small grazers, particularly copepods, amphipods, and krill. These crustaceans feed directly on ice algae, either by filtering cells from the water, scraping them from ice surfaces, or, in the case of very small copepods, entering brine channels to feed within the ice. The efficiency of energy transfer at this step is relatively high, perhaps 10 to 30%, meaning that a substantial fraction of algal production gets converted into grazer biomass. This high efficiency reflects the quality of ice algae as food. They are rich in essential fatty acids and nutrients that support rapid growth in consumers. Grazer populations can reach remarkable densities. Arctic copepods aggregate at the ice-water interface during spring blooms, with abundances reaching tens of thousands per cubic meter of water. Ice amphipods carpet the underside of ice floes in some areas. Antarctic krill form swarms so dense that the water appears to boil with their movement. These high densities represent the conversion of microscopic algal biomass into larger packets that are more accessible to the next trophic level. The pathway from grazers to planktivorous fish represents another crucial link. Fish like Arctic cod and Antarctic silverfish feed primarily on small crustaceans, consuming copepods, amphipods, and juvenile krill. These fish occupy a critical position in ice ecosystem food webs because they transfer energy from the small, dispersed grazers to larger predators that cannot efficiently capture tiny prey. Fish aggregate zooplankton biomass spatially, schooling in areas where prey is abundant, and they grow to sizes that make them suitable prey for seabirds, seals, and whales. From planktivorous fish, pathways diverge to multiple apex predators. Ringed seals in the Arctic feed heavily on Arctic cod, consuming hundreds or thousands of fish over the course of a year. A female ringed seal must consume enough cod to support not only her own metabolism, but also the production of milk to nurse her pup. Polar bears, in turn, prey on ringed seals, specializing particularly on pups born in spring. The energy that began as sunlight captured by ice algae, transferred through copepods to cod, has now reached the apex predator of the Arctic. Seabirds create another pathway from fish to higher trophic levels. Guillemots, moorés, and other diving seabirds pursue Arctic cod and other fish beneath the ice, capturing them in fast-paced underwater chases. A breeding seabird might bring dozens of fish back to its chick each day, transferring the energy from marine to terrestrial systems when the bird returns to its cliff nest. In the Antarctic, penguins serve similar roles, diving beneath ice floes to capture silverfish and krill, then returning to their breeding colonies to provision their chicks. Not all energy flow follows these classical grazing pathways from algae to herbivores to predators. An important alternative pathway runs through the microbial loop, where dissolved organic matter excreted by algae or released from cells is consumed by bacteria. These bacteria are then eaten by heterotrophic protozoans, which in turn become food for small metazoans. This microbial pathway processes a substantial fraction of primary production, perhaps 20 to 50% in some systems, though with lower overall efficiency than the direct grazing pathway. Detritivores and decomposers form another essential component of ice food webs. Dead algae, fecal pellets from grazers, molts from crustaceans, and other forms of particulate organic matter accumulate in the ice and fall into the water below. This material supports populations of deposit-feeding organisms on the seafloor beneath the ice, including worms, mollusks, sea cucumbers, and brittle stars. In shallow Arctic seas where light reaches the bottom, seafloor communities can be remarkably productive, rivaling tropical coral reefs in biomass, though not in species diversity. The benthic community beneath sea ice creates a connection between the ice ecosystem and the seafloor that closes nutrient cycles. When organic matter sinks from the ice to the bottom, it carries nutrients with it. Bottom-dwelling organisms process this material, releasing nutrients back into solution through their metabolism. Mixing and upwelling can eventually transport these regenerated nutrients back to the surface where ice algae can use them again. This vertical coupling between surface and deep communities allows the ecosystem to maintain productivity over time. Temporal dynamics add complexity to ice food webs. The seasonal pulse of algal production creates a boom and bust pattern, where food is superabundant during the spring bloom but scarce during winter. Animals must either synchronize their life cycles with this pulse, timing reproduction and growth to coincide with the bloom, or they must develop strategies to survive the lean periods. Many copepods accumulate large lipid reserves during spring feeding, then descend to depth where they metabolize these reserves slowly through the winter. Seals likewise accumulate fat during summer feeding, drawing on these reserves through winter and while nursing pups in spring. The match or mismatch between the timing of primary production and the requirements of consumers profoundly influences ecosystem functioning. If warming temperatures cause ice algae to bloom earlier in the spring, but grazer life cycles remain synchronized with photoperiod rather than ice conditions, grazers may miss the peak of algal production, reducing their reproductive success and cascading through higher trophic levels. Such phenological mismatches are a major concern, as climate change alters the timing of seasonal events. Food web complexity provides resilience, but also creates vulnerability. When multiple pathways exist for energy transfer, the loss of one pathway can be compensated by others. If amphipod populations decline, copepods might fill some of the same grazing roles. If Arctic cod becomes scarce, seals might switch to other prey species. This redundancy helps buffer the ecosystem against perturbations. However, the loss of keystone species that lack functional replacements can trigger cascading effects. The decline of sea ice threatens species like polar bears and ice seals that have no alternative habitat, potentially causing their loss and the unraveling of food web connections they maintain. Energy flow analysis reveals inefficiencies but also explains biomass patterns. At each trophic transfer, typically only 10 to 20% of energy passes to the next level, with the rest lost to respiration, excretion, and unconsumed production. This means that it takes roughly 10 kilograms of algae to produce 1 kilogram of copepods, 10 kilograms of copepods to produce 1 kilogram of fish, and 10 kilograms of fish to produce 1 kilogram of seal. The pyramidal structure of biomass in ecosystems reflects these transfer efficiencies, explaining why apex predators are always rarer than prey species. Yet, sea ice ecosystems can support remarkably high abundances of top predators compared to many other systems. The high productivity of ice algae blooms, the efficient grazing by crustaceans, the strong aggregation of biomass by schooling fish, and the temporal concentration of resources during the spring bloom all contribute to sustaining large populations of seals, polar bears, whales, and seabirds. The seasonal pulse of productivity, while creating challenges, also creates opportunities for predators to access concentrated prey resources during critical periods. The food web perspective also highlights connections beyond the ice itself. Ice-breeding seals give birth to pups on the ice, but these seals hunt in the water below, creating a link between ice surface habitat and pelagic food webs. Seabirds nest on terrestrial cliffs but forage in ice-associated waters, transferring nutrients from sea to land through their guano. Polar bears hunt on ice but den on land, and in summer, they may supplement their seal-based diet with terrestrial foods including vegetation, bird eggs, and caribou. These cross-habitat connections embed sea ice ecosystems within larger regional food webs that span marine and terrestrial realms. Contaminants follow food web pathways just as energy does, but with an important difference. Many contaminants biomagnify rather than diminishing with each trophic transfer. Persistent organic pollutants like PCBs and heavy metals like mercury accumulate in organisms and become more concentrated at higher trophic levels. Polar bears and seals thus carry contaminant burdens far exceeding those in the algae and fish they ultimately depend upon. This biomagnification makes top predators particularly vulnerable to pollution, even in the remote polar regions far from contaminant sources. Understanding sea ice food webs requires integrating knowledge across scales, from the biochemistry of individual cells to the behavior of apex predators, from microscopic bacteria to the largest whales, from seasonal rhythms to multi-year cycles. This integrative perspective reveals that the sea ice ecosystem is not a collection of independent species but rather a network of relationships where each component influences and depends upon many others. The health of the whole system depends on maintaining these connections, and disruption at any level has the potential to propagate through the entire web. Climate change and the shifting ice. The sea ice ecosystems we have explored throughout this journey are not static museums of polar life, preserved unchanged through millennia. They are dynamic systems that have always varied with natural climate cycles, expanding and contracting, thickening and thinning in response to atmospheric and oceanic conditions. But the changes now unfolding exceed anything experienced during the period when modern sea ice ecosystems evolved. Climate warming, driven by human greenhouse gas emissions, is fundamentally restructuring polar environments, with consequences for sea ice and the organisms that depend on it that we are only beginning to understand. The physical changes to sea ice are dramatic and well documented. Arctic sea ice extent at its September minimum has declined by more than 40% since satellite measurements began in 1979, a loss of roughly 3 million square kilometers of ice habitat. The decline is even more pronounced for multi-year ice, which has decreased by more than 90% in extent and volume, transforming the Arctic Ocean from a landscape dominated by thick, stable ice to one dominated by thin, seasonal ice. The rate of decline has accelerated in recent decades, with the 12 lowest minimum ice extents on record all occurring since 2007. The thickness of Arctic sea ice has decreased as dramatically as extent. Submarine measurements show that average ice thickness in the Central Arctic Ocean has declined from approximately 3.6 meters in the 1980s to less than 2 meters today. The loss of volume that results from combining declining extent and thickness is staggering, representing a reduction of perhaps 70 to 80% in total Arctic sea ice volume. This means that even when ice is present, it is thinner, more fragile, and more susceptible to melting than the ice that once filled the Arctic. The timing of seasonal ice formation and breakup is also shifting. Spring melt now begins earlier, and autumn freeze-up occurs later, extending the ice-free season by weeks or months in some regions. This change in timing profoundly affects ecosystems because many organisms have evolved life cycles synchronized with historical ice phenology. When ice algae bloom earlier, but grazers reproduce on a schedule set by photoperiod rather than ice conditions, mismatches occur that can reduce energy transfer and reproductive success. Antarctic sea ice has shown more complex patterns, with regional variations that defy simple generalizations. The Antarctic Peninsula has experienced rapid warming and dramatic ice loss over recent decades, with glaciers retreating and ice shelves collapsing. In contrast, some regions of East Antarctica saw modest increases in sea ice extent from the 1970s through the early 2010s. However, since 2016, Antarctic sea ice has shown unprecedented volatility and decline, with record low extents that suggest the Antarctic may be entering a new phase of rapid change, similar to what has occurred in the Arctic. These physical changes to ice translate into biological impacts that cascade through ecosystems. The most direct effects fall on organisms that require ice as physical habitat. Polar bears depend on sea ice as a platform for hunting seals. As the ice-free season lengthens, bears spend more time on land where they cannot hunt efficiently, forcing them to fast for longer periods. Body condition declines, particularly in females who must accumulate fat reserves to support pregnancy and nursing. Reproductive success decreases as fewer cubs are born and survival of those born declines. Similar impacts affect ice-breeding seals. Ringed seals require sufficient snow depth on stable ice to construct birth layers where they nurse pups. Warming temperatures lead to more frequent rain-on-snow events that create hard, icy surfaces difficult for seals to excavate. Earlier ice breakup can disrupt nursing before pups are weaned. These factors combine to reduce pup survival, and multiple populations of ringed seals have shown declining trends. Other ice-dependent seals, including bearded, harp, and hooded seals, face analogous challenges as ice habitat degrades. Effects on ice algae communities are more nuanced. Reduced ice extent means less total habitat for ice algae, potentially decreasing overall primary production by this component of the ecosystem. However, thinner ice allows more light penetration, which could increase productivity per unit area. The net effect depends on whether the productivity gains from thinner ice offset the losses from reduced extent, and current evidence suggests that in many regions, the extent reduction dominates, resulting in decreased total ice algae production. Changes in ice algae affect all organisms that depend on them. Copepods that migrate to feed on ice algae in spring may find less food available, reducing their reproductive output and abundance. Amphipods and other ice-associated grazers decline as their habitat shrinks. Arctic cod, which rely on ice for juvenile habitat, and whose prey consists largely of ice-associated zooplankton, show population changes linked to ice conditions. The effects propagate upward through food webs to seabirds, seals, and ultimately polar bears and humans who depend on these resources. The community composition of ice ecosystems is also shifting. Warmer temperatures allow temperate and boreal species to expand northward into traditionally Arctic waters. Atlantic cod are now found in areas once dominated by Arctic cod. Temperate zooplankton species extend their ranges poleward, potentially competing with or replacing Arctic specialists. Even phytoplankton communities are changing, with smaller species adapted to warmer, more stratified conditions replacing the large diatoms that once dominated Arctic blooms. These biological invasions create novel ecosystems with no historical analog, communities composed of species that never before interacted. The consequences are difficult to predict. Will the newcomers fill ecological roles vacated by declining Arctic specialists, maintaining ecosystem functioning? Or will they create different, less productive ecosystems? Will food web connections remain intact as species compositions change? These questions remain largely unanswered, though early evidence suggests that novel communities may be less productive and less able to support traditional harvest species important to Arctic peoples. The timing mismatches mentioned earlier represent another critical impact. Many Arctic organisms time their reproduction and growth to coincide with the peak of the ice algae bloom. If warming causes the bloom to occur earlier in the spring, but organisms cannot advance their life cycles to match, they miss the peak food availability. This has been documented in some copepod populations, where reproduction now occurs after the algae bloom has passed, reducing reproductive success. Such phenological mismatches can propagate through food webs, affecting all species that depend on the affected organisms. Ocean acidification adds to climate stresses. As atmospheric carbon dioxide increases, the ocean absorbs more CO2, forming carbonic acid that lowers seawater pH. This makes it more difficult for organisms to build shells and skeletons from calcium carbonate or silica. While polar waters have naturally low pH due to cold temperatures enhancing CO2 solubility, further acidification threatens ice algae that build silica frustules and other organisms with mineralized structures. The combination of warming and acidification may create conditions outside the tolerance range of many polar species. Climate change also affects sea ice ecosystems indirectly through impacts on ocean circulation. The Atlantic meridional overturning circulation, which brings warm water northward and influences ice formation in the Arctic, shows signs of weakening. Changes in the Antarctic circumpolar current affect ice dynamics and biological productivity around Antarctica. These large-scale circulation changes can alter nutrient delivery to surface waters, modify ice formation patterns, and shift species distributions in ways that compound direct warming effects. Despite these concerning trends, some organisms show remarkable resilience. Many polar species have evolved in environments characterized by high variability and have broad tolerance ranges. Some populations appear to be adapting to changing conditions through behavioral plasticity or evolutionary change. Polar bears in some regions have shown dietary flexibility, supplementing or replacing seals with terrestrial prey. Some seal populations are shifting their distributions or timing of reproduction. Ice algae may adjust their phenology to track changing ice conditions. The challenge is that rates of change now exceed anything these organisms have experienced during their evolutionary history. Adaptation takes time, requiring multiple generations and substantial genetic variation in traits under selection. Rapid change may not allow sufficient time for evolutionary responses, leading to population declines or local extinctions. Even species showing current resilience may face thresholds beyond which they cannot adjust, tipping into rapid decline when critical limits are exceeded. Human responses to these changes will partly determine their ultimate impacts. Reducing greenhouse gas emissions can slow warming and provide more time for ecosystems to adjust. Protecting remaining high-quality ice habitat can serve as refugia for ice-dependent species. Managing fisheries sustainably can avoid compounding climate stress with overharvest. Supporting indigenous communities who depend on marine resources can help maintain traditional practices while adapting to new conditions. International cooperation, particularly through the Arctic Council and the Antarctic Treaty System, provides frameworks for coordinated action. Yet, we must acknowledge the reality that substantial additional warming and ice loss are already inevitable due to past emissions, and that even aggressive mitigation will not prevent significant ecosystem changes. Sea ice ecosystems are being transformed before our eyes, shifting towards states that have no historical precedent. Some species will decline or disappear. New communities will form. Productivity may decrease in some regions while increasing in others. The iconic polar ecosystems that have captured human imagination for centuries are entering an uncertain future, and we are witnessing changes that will reshape these systems for millennia to come. Closing reflection. As we reach the end of our journey through sea ice ecosystems, let yourself settle into a quiet space of reflection. We have traveled together from the microscopic scale of brine channels to the grandeur of polar bears and whales. We have witnessed adaptations of breathtaking elegance. From algae photosynthesizing in twilight conditions to seals maintaining breathing holes through meters of ice. We have explored food webs of remarkable complexity, seasonal rhythms of dramatic intensity, and geographic variations that remind us of life's endless creativity. And we have confronted the sobering reality that these extraordinary ecosystems face unprecedented challenges as our changing climate reshapes the polar regions. The sea ice ecosystems we have explored represent something precious and irreplaceable. They evolved over millions of years, shaped by the unique conditions found only at Earth's poles. The organisms that inhabit them possess adaptations found nowhere else, solutions to biological challenges that expand our understanding of what life can achieve. The productivity they sustain supports not only the iconic species that capture our attention, but entire webs of life extending from microscopic bacteria to the largest animals ever to exist. And they play roles in Earth's climate system that extend far beyond their frozen boundaries, influencing ocean circulation, atmospheric chemistry, and the planetary energy balance. These ecosystems also hold meaning that transcends their ecological importance. They represent wildness, places where natural processes still dominate and human influence, while growing, has not yet completely overwhelmed the ancient rhythms of polar life. They inspire wonder, reminding us of our planet's diversity and beauty. They challenge us, demonstrating that life thrives even in conditions that seem impossibly harsh. And they humble us, revealing how much we have yet to learn about the natural world. The changes now unfolding in polar regions should concern everyone, not just scientists or people living at high latitudes. Sea ice ecosystems are early warning systems, sentinels experiencing climate change impacts before they become apparent elsewhere. The transformations occurring in ice-covered seas foreshadow changes that will eventually affect all of us. The loss of sea ice alters the planetary albedo, reducing Earth's reflectivity and accelerating warming. Changes in ocean circulation, driven partly by ice melt, affect weather patterns globally. The decline of polar species represents losses of biodiversity that diminish all humanity's natural heritage. Yet this journey through sea ice ecosystems should inspire not just concern, but also resolve. Understanding these systems reveals what is at stake and what we stand to lose if we fail to act. It highlights the urgency of reducing greenhouse gas emissions, protecting critical habitats, and supporting the resilience of both ecosystems and human communities. And it demonstrates that knowledge itself is a form of action, that understanding complexity is the first step toward developing solutions. If you have found value in this peaceful exploration of sea ice ecosystems, I invite you to like this content and subscribe to the Sleepy Science Society. Your support helps these journeys continue and allows other curious minds to find this corner of calm science education. Share this with others who might appreciate learning about the hidden wonders of our frozen seas. Let your interest in these remarkable ecosystems inspire conversations, questions, and perhaps actions that contribute to their preservation. For now, simply breathe deeply and let the knowledge you have gathered settle into your thoughts. Imagine the golden brown carpets of ice algae photosynthesizing in twilight beneath the frozen Arctic Ocean. Picture the intricate network of brine channels threading through the ice, each a microscopic river of life. See the amphipods grazing upside down on algal forests, the Arctic cod schooling in the shadowed water below the ice, the seals resting on the surface while their pups learn to swim. Hold these images as you drift toward sleep, and let them remind you of the extraordinary complexity and beauty that exists in places most humans will never visit. The sea ice ecosystems we have explored tonight continue their ancient cycles, indifferent to whether we pay attention or not. They do not need human appreciation to exist. Yet they are vulnerable to human impacts in ways we have only recently come to understand. Our choices in the coming years and decades will determine whether these ecosystems persist, adapt, or disappear. That responsibility is sobering, but it is also empowering, because it means we still have the opportunity to make a difference. Rest now in the knowledge that you have glimpsed something remarkable. A world of frozen seas that harbor life of astonishing diversity and resilience. Let your breathing slow. Allow your thoughts to quiet. Let the wonder of sea ice ecosystems accompany you as you drift toward sleep. And perhaps when you wake, carry that wonder with you into the waking world, where knowledge can become action and curiosity can become care. Good night.